In question 6, I wanted to use the sequence of the Y chromosome, and I wanted to annotate that sequence in terms of where are there possible genes here, and you're going to do a gene prediction using an ab initio program, which means we're going to use a software that looks at that DNA sequence and looks for the cues for where are there possible genes in here. So the program that I used for my PhD was called FGenesH. Let's see. And if you don't remember the name of the program, you could Google gene prediction software. There is a bunch of them. At that time, I think that was one that performed well for my purpose. But here, you can look at, there's a bunch of them. And I'm only going to show you one out of all of these for obvious reasons. I think this Augustus now it's like considered better maybe, but anyway, we're going to use this one, the HMM based, that's called the hidden Markov model. I think that's what that H is for. So we're going to use this gene software, this software that predicts gene for our purposes. Could you use any of the other ones? Yes, but then it's on you to learn what their output is telling you. And maybe you're going to work in a lab and they use GeneMark or anything. So I think a lot of people use this now. But we're going to use this one. So I'm just going to copy that and search FGenesH. And well, let's see. I try not to click on the add ones. We're going to go to this thing here. And then oh, okay, let's see. I just want to predict genes. Structure, no, that's not the one. And not bacterial. We have the Y chromosome in our case. Okay, here, annotation of animal genomes. So let's see. I also like this page so that you can see all kinds of stuff that people do. But we are doing annotation. We're doing this gene identification. It's really gene prediction. We don't actually know it's a gene there. We're just using the software to get our best guess of where it is. And we're going to use the HMM one. And we have the Y chromosome, so we're going to do animal. And now there are more choices. We're going to do, let's do this one. So I guess this is, this is like an update to that. I'm just going to use that first one, just FGenesH. Good. So in here it says paste your DNA sequence. And I'm going to use the lazy approach, which is I'm just going to paste the DNA sequence. It might give me an error, which it has given me in the past, or maybe they changed the software now to do some of the work for us. So here's what we're trying to do. There is that 42,000 plus nucleotide sequence of the Y chromosome, and the accession number was this. And I could just Google that got to the same place as it did in a previous question. So we'll do that. And maybe I don't remember the acronym NCBI. So, but if you do remember the acronym NCBI, that's like the hardest part to remember. And you can just Google that or Google this whole thing, and you're going to get to the page where you can have a drop-down menu and say, I want the sequence of this thing. So I'm going to Google that because it was the shortest way of getting there. And I click here, and there's the whole Y chromosome, and I put how many nucleotides in the beginning so that you know you have the right sequence. Is a little over 42,000. So all I'm doing now is scrolling down to get to the end of this and copy that. And I'm going to paste it here and cross my fingers. Oh, let's see, organism. It is humans. So this thing here is what did they use to train the software? And it has to do with which cues are common in the promoter region. So that's why you'd have like different possibilities here. And then do search. And it says error. No, I already knew it was gonna do that. So let's see. And here's where our result should have been. But okay, I wish they would update it so that I don't have to do some pre-editing before I can submit the work here. Okay, so it did what I thought it was going to do. What it's complaining about is the fact that there are things here that I entered that are not AT, Cs, and Gs. They're numbers. And so you're just going to Google remove numbers or something like that. So before I just Googled remove numbers from a DNA sequence. So let's Google that. Remove. You can say non-ATG characters, but anyway, from DNA sequence. Whatever does it for you. Let's see. I often like things that are housed in this domain, bioinformatics.org. So that's my reason to pick that one, because I've used this before. The other one probably would work the same way. Okay, so I pasted the whole sequence there. And here, this is what's going to do. It's going to remove everything that is not a G, A, T, C, or N. And you're like, N? Yeah, sometimes there are N's in here on the DNA sequence, which is when during a sequencing round, there wasn't a clear peak. I don't know if you remember the chromatogram. Anyway, the computer puts an N, saying it's in some nucleotide here. I don't know which one. And remove characters with nothing. Yeah, I don't want any spaces. Don't change the case. I don't care about that. Okay, so submit. Ta-da! My goodness, how I like when the computer does the work for me. All right, so I'm going to highlight and copy all this. So Command C. Well, I guess the keys I mostly strike are the Command C, Command V, Command F. No, I have to find all the time. I don't want to read the whole page often. Okay, so here I'm going to do Command A for like all, to delete all that. That was the one that had the numbers. And now I'm going to paste my new sequence. It's the same sequence, just without numbers. Let's see if I don't actually select organism, if it will do something different. Search. It probably has something as default. But let me show you that the error message was not because I chose human. Okay, so now I have the sequence without any numbers. And if I don't say anything, you could just leave the default as whatever it was there. I'm only running it with humans now to show you that the error was not because of this selection. It was really because of the numbers. And then I love when it does what I said it's gonna do. So there are two ways you can look at the output. And here's one way. I'll go over it. The other one is we look at pictures. I think it's easier to understand it through pictures. So yeah, let me look at the pictures first, and then I'll go back and show you where these elements are in the text-only file. So I didn't write it in sequence. This would be whatever name I gave it. And here's how long, how many letters I entered in the search box. And here's saying how many genes does this software predict. So the whole Y chromosome, it's saying there are 2 genes predicted. And then it says, in the plus chain, 2. In the minus chain, 0. Okay, so this is, bioinformatics. It's interplay between computer scientists and biology people. So somebody decided to call plus chain. All that it is saying is in the sequence you entered, there were 2 genes predicted. If the computer took the sequence you entered, reverse complemented it, then in that reverse complement, when it looked for genes in the reverse complement, which is that minus chain, then in this case, there were 0. So that 2 and the 0 are the things that change, but the computer and that little 2 here, but the computer is always going to say number of predicted genes, however many, and it's going to be the addition of how many genes in the sequence as you entered it. So it would be, so the plus chain is considering that the DNA sequence you entered is the sense or the coding DNA strand. And the minus chain would say the sequence you gave me is the template strand, and the reverse complement of that is what contains the mRNA. But in this case, it looks like there's nothing on the other strand. Most chromosomes will have some genes in one strand, other genes in the other strand. It's not always like one strand of the whole chromosome is coding and the other one is not. They can alternate, not necessarily, but they can. Okay, then predicted exons, there are 10 of them, all 10 in those 2 genes, and nothing in the, because there is no gene predicted on the reverse complement of the sequence I entered. And then, all right, positions, never mind about that one. I'm going to talk about the main aspects here. All right, so these things here, CDS is coding sequence. It is the DNA sequence that, okay, it becomes the mRNA sequence, which then encodes an amino acid. So the coding sequence is the exons, okay? And whatever is not coding sequence can still be part of the gene. Remember that there are introns, there is a 5' UTR, there's a 3' UTR. So let's go by parts. Coding sequence, the nucleotides that will end up as amino acid in the protein. And for this specific tool, the F is for first. So it's our first exon. The gray things, the I is for internal. So that's some exon that is neither the first nor the last. So imagine some protein has 3 exons, then this would be your first exon, this would be your second exon, and this would be your third exon. If it had 4 exons, you would have one of that, 2 of these, and 1 of this. So there's always just 1 of this and 1 of this, and however many in the middle, if you have more than 2. Some genes don't have introns. And you're like, oh, you're talking about prokaryotes. I'm like, oh, I'm glad you're thinking about that. But even eukaryotic genes, sometimes they don't have introns. So in this case, there's a coding sequence only. So it's saying it doesn't look like this gene has any introns. So that's the case of this one here. So it looks like this whole thing is 1 exon. So no introns for this gene. It happens. And then, okay, the green thing is the poly A tail. That is the signal to put a bunch of little A's here. So when transcription ends, then it will end here, and part of the processing of the mRNA is put an A in the end. It adds to the stability of the mRNA in the cell. So from perspective of gene prediction, the computer is looking for cues that will say that that's the end of transcription. So not of translation. This is not the stop codon. It's the end of transcription. So that's the signal to enter the poly A tail. That's the end of transcription. And then this TSS is the transcription start site, not translation, not the start codon, not the ATG or EUG. This is where the mRNA starts. So not where the protein that comes out of the mRNA will start. Okay, so here, let's look at each of these genes. The first gene predicted starts in nucleotide number 525, ends in nucleotide number 2603. So the mRNA goes, so if I ask you how long is the mRNA is from here to here. So it's that difference plus 1, because that 525th nucleotide is nucleotide number 1. It counts. So the length of the gene is that. It's from the poly A signal to the transcription start site. So that is how long this gene is. But how long is the protein? Oh, the protein is only what comes out of the exons. In this case, this has only one exon. So the protein will be this many nucleotides divided by 3, because every 3 nucleotides encode 1 amino acid. That's how long this protein would be. Now, how do you know how many nucleotides are there in this exon here? So I'm glad you asked. So here, this part here is how long the exon is. It actually does the math for you over here. And I'm going to skip the scoring part. So this exon goes from nucleotide number 1684 to nucleotide number 2592. So, which is a little bit before the poly A tail signal that is in 2603. So this has a very short sequence between the end of that exon and the end of the mRNA, so where they put the poly A tail. So I told you in class what those things are called, but here, let me remind you. The gene is everything, that's the DNA sequence that gets transcribed. The part that gets transcribed is from the transcription start site to the poly A tail signal. And then in here, you would have exons, and in some cases you have, so an exon, really remember that the first exon, so that's the little thing there. That's a very short exon, very short last exon, and then a bunch of exons in the middle. So the red, the blue, and these grays, those are the nucleotide sequences that will, from the mRNA, that will end up as amino acids in the protein. And then the sequence between the transcription start site and the start codon, which is so like in here and in here in the red. So that chunk there is your 5' UTR. And then, which is very short for this other gene. And this chunk here, this is where translation stops, and this is where transcription stopped. So there is this piece of the mRNA that is even before the poly A tail. There's this piece here that did not get translated. So this is your 3' UTR. If I ask you how long are those parts, then the information comes from the numbers that are like over here. So the transcription start site is in nucleotide number 2802. So there. Okay, our first exon, so coding sequence first, is, so this is how long that exon is. If you want to, the computer did the math here. There's a small difference. So that's about 28 nucleotides. And then how long is the 5' UTR here? So the space between the triangle, the blue triangle and the red triangle, it's where the transcription start site starts and your first exon starts. So you subtract those to get how large is your 5' UTR. And down here are the sequences for each of those two predicted genes. So, and the program splits it into the exons. So the first gene had only one exon. Here is that first exon of that gene, and or ends here. And now it's the second gene. No, wait, what's this one? Okay, now I have to look. But here's the, oh, wait, so here's the mRNA of that gene. And then here is the translation of that gene. And then over here is the mRNA of the second gene. And then what's the translation of that second gene is over here. And all the things that I just showed you are also in the just text, but I feel like the drawings really help